Next, we're gonna have Dr. Venditti, who is coming to us from the National Institute of Health. He runs the research there for organic acidemias, and propionic acidemia is one of the organic acidemias that his team has been studying for many years. And he's going to share with us some research updates and the approach to finding new therapies for propionic acidemia. Dr. Venditti.
Okay. All right, can everybody hear me? Yeah, okay, let me just, this seems like a simple advance. Okay, well, first of all, thank you, everybody, for coming this morning, and thank you for inviting me. It's an honor to come and speak on something I've been deeply interested in since 1999. The reason 1999 is the date I mentioned is because that's the day I started my fellowship at the Children's Hospital of Philadelphia. And at that time, newborn screening for propionic acidemia was just beginning in the state of Pennsylvania. The previous slide Dr. Yama showed had 2003. That's when the United States government recommended that all the states screen for propionic acidemia, and as well as other conditions. But in 1999, Pennsylvania started a pilot study, and I started meeting families, many families who had a lot of different metabolic conditions, organic acidemias, of which propionic acidemia is one. And I was seeing an impact of screening because across the river from the Children's Hospital of Philadelphia is New Jersey, and they were not screening for the disorders. And they weren't screening for all the conditions in every hospital in the state of Pennsylvania. I became really interested in this because one of the tenets of newborn screening is that a condition must have, amongst other things, a treatment. And so there was a lot of discussion in the academic circles and the public policy circles about screening for metabolic disorders in general. And it goes like this. It's a simple discussion. If you can't treat something, why would you screen for it? This is the mentality some people have. So that is how I got interested in organic acidemias, particularly MMA and PA, because I was meeting lots of families very early in life, the first week of life, when we got the screen result that indicated that they had one of those disorders. So I'm going to talk today a bit about some of the things we've done on propionic acidemia. And first to acknowledge the wonderful efforts that Dr. Helmus, you've done here in the clinic and how amazing this resource is. I'm showing you a picture of a great group of people. This is the team I work with at NIH. I won't be able to introduce and show everyone's picture on there. You can see their pictures, but Dr. Shalachkov, who some people may know, that's Oleg. He's sort of in a high and mighty position. He was in the group and started our clinical research program on propionic acidemia. He's since moved on, and Dr. Carolina Galarreta-Ema is now one of the physicians in the group that's leading the PA clinical studies. And also on the team, it's hard for me to sort of twist my head around and see her, but we have a group of people. We work on gene therapy, we work on informatics, we work on animal models, we work on new therapies. And so everybody in the group, many people there are working on propionic acidemia, and particularly Randy Chandler. I see Randy in the middle there, and Lena. Lena's not in this photo, but so a lot of the team works on both PA and MMA. I don't have any active corporate partnerships. I should also say I'm an inventor. I have a number of patents. I don't actually have the patents. The NIH has them, but it's a disclosure in case anybody wants to look up the patents. There's the application numbers. So today I was going to try to present a bit about like the research approach to studying a metabolic disorder, propionic acidemia. I'm going to give a bit of background, and I've been fortunate to follow other speakers, so I'm not going to go through this in great detail. And then I'm going to focus on sort of three vignettes. One is what's called natural history study. The next is something called biomarkers. And the last is going to be on gene therapeutics. Okay. So this was mentioned in a beautiful overview the inborn errors of propionyl-CoA metabolism, of which propionic acidemia is one of them. There's many of them. They were originally designated as ketotic hyperglycemia. That's a non-specific biochemical finding that's actually shared by PA, MMA, isovaleric acidemia, beta-ketothiolase deficiency, et cetera. There's many. But that was the first step of recognizing PA as a metabolic disorder, and that was in the 60s. It was eventually noticed and proven that patients that had this so-called ketotic hyperglycemia had massive elevations of organic acids. So they didn't have a glycine problem. That's an amino acid. They had an organic acidemia, and this has happened in the 60s and mainly in the 70s. Most patients that have this have either MUT-MMA or propionyl-CoA carboxylase deficiency, and that causes propionic acidemia when you don't have that enzyme. As a group of disorders, patients that have these conditions are very, very difficult to treat because the only treatments we currently have are diets and cofactors. And when the patients still develop complications on the current regimens, that indicates that the current regimens need to be improved. And they're prototypical for a large group of disorders. When I say prototypical, I mean they sort of represent many conditions called organic acidemias, of which there are many, many, dozens and dozens. This is a very hard thing to go over here without a pointer. But what this little cartoon shows is sort of a real breakdown of metabolism that was gone over early in the first talk, showing the location of the propionyl-CoA carboxylase enzyme. And that's that, you look on the screen, it'll be on your upper left, that little cartoon box that has PCC-A, B, and biotin, okay? Those are the subunits of the enzyme propionyl-CoA carboxylase. So if patients that are in the Plains community predominantly have a change in the PCCB part of this enzyme. But in the entire population, they're about 50-50. So if you had a group of 100 patients and you didn't have any, so randomly selected, half would have PCC-A, half would have PCCB type, and all different changes in the gene. Okay, so they're very different, and that is really important when you try to figure out mechanisms and develop treatments. In terms of the therapies, we talked a bit about this. The mainstay of treatment that was instituted in 1960 was protein restriction, and it had a positive effect on the patient. So we still do that, protein restriction. We give carnitine for a specific reason. And in some patients, again, the main symptoms we're trying to block in the patients overall as a group is the propensity for metabolic decompensation and high ammonia. And that can happen recurrently. That comes from the liver. So that's why people do a liver transplant. It's not because some of the patients maybe you've heard about or read about, they don't have liver failure. They're getting a liver because we're just giving all new, brand new enzyme into their liver to make them stable. Patients can have kidney disease, and I'm going to talk about that. Some patients require a liver and a kidney transplant. And some patients have required liver and, as we heard, heart transplants. And one of the very first patients I met, the recommendation was for a liver, kidney, heart transplant. Now, not every patient needs this. But when we think about a transplant, you have to think, as a community, scientifically, we think, is there a better way than a transplant? And the answer is yes. So when we look overall and put PA and MA and the cobalamin disorders together, they go into a group. They're very closely related. That's why they're all together on the slide. And you can see, if you look here and read, the different symptoms and signs that occur in the different organs in a patient. So each patient can have a unique set of complications. In the Plains community, I understand that it's mainly the cardiac problems, but some patients I heard can have the metabolic instability. So they can, if they're not known to have this, they can go to the hospital with sick, vomiting, ketoacidosis, and be very, very sick and need IV fluids. Whether they get hyperammonemia, high ammonia, I don't know that. Most patients with these disorders share this common feature of metabolic instability. That emanates from the liver. So that's, again, going to reinforce why people elect, in some cases, to give a liver transplant. Now, if the kidney doesn't work, There's no medicine that you can take that can replace that. Eventually, that requires one or two things. Renal replacement therapy is either dialysis or a kidney transplant. Now, not every patient gets this. And then, of course, the cardiac complications, which, as we heard about from our previous speakers, are a bit mysterious. And I'm going to just take a second to come back to the topic from that Dr. Meisner brought up about why is that. And there's a model that we published that I believe is relevant, which postulates something called conditional autonomy as the pathophysiologic mechanism for why the heart is affected in PA and can be disparate from the heart involvement. And of course, the conditions are detected by newborn screening. And I learned something very interesting today, that 50% of the patients in the Plains community are missed by newborn screening. So this is really urgent to figure out a new way to get to those patients very early in life. So in terms of how to study an organic acidemia PA in this case, the approach that we've adopted over the years is to perform clinical research and then complement that with animal modeling. So it turns out animals have the same types of genes, many do, that humans have. In fact, one of the earliest studies I ever did, we used roundworms, little tiny worms called Cenorhabditis elegans. It turns out these little tiny worms have all the types of genes that humans have for propionyl-CoA metabolism and cobalamin metabolism. We tend to use mice because we can genetically engineer mice. It's all ethical and it's routine to do. The other types of animals we study in the laboratory are zebrafish. You might see them in the store. Well, it turns out zebrafish also have all the same genes humans do for using cobalamin and propionyl-CoA metabolism. And we've made zebrafish models of a number of organic acidemias to study them. And then, of course, we use high-throughput sequencing and human genetic methods to help inform what we do. And by doing this, we can try to figure out what the disease mechanism is, and then develop a new treatment. And then see if something we see in a mouse correlates to a patient, or vice versa, if something we see in a patient correlates to a mouse or a zebrafish. So I'm going to warn people there's going to be a little bit of technical details coming up here that we can discuss after the break. I'm going to try to simplify the approach. Where do we do the research? This is a picture of the National Institutes of Health. This is the world's largest clinical research hospital. It's located in Bethesda, Maryland. Every single bed in this hospital is dedicated to research. And this is where we see patients. To put it in perspective of the size of this, if you've ever seen a golf course, this campus was once one half of a golf course. The other half, which is across the street, is the military medical complex for the United States of America. So this is a very massive hospital. And I moved here right from the Children's Hospital of Philadelphia to start the studies. Here's some statistics for people that are interested. We have 200 inpatient beds. We about have 100,000 visits a year. These are pure research. And we have hundreds of clinical trials. Many are for rare disorders that sometimes don't have a lot of corporate interest. All right, that's a bit about the background. Now I'm going to talk about natural history. Like, we keep hearing about natural history. What is a natural history study? So this is a definition. I think this is a good definition. It's not, people could, argue about this. What is it? It's a clinical research study that tracks the progression of a disease or medical condition over time without any experimental interventions or treatments. So obviously there's a limitation there, because basically everything that's done on patients with PA, you have a dietary prescription, you take a cofactor. The goal of this is to understand how the disease develops, behaves, and affects individuals in the natural state. And the studies are really valuable to provide insights into the typical course of the condition, from its onset through the progression, and include data like symptoms, complications like heart problems, and outcomes, including survival rates. So the key aspects include some of the following. Observation over time. No experimental treatment. So this is really important. If someone comes into a natural history study and they've been a lot of experimental therapies and then something happens to them, how do you know it wasn't from the experimental therapy, right? So this is part of the fundamentals of what you tried to do. You need to have comprehensive data collection. And this is actually fairly difficult because if it's a rare patient population and people want to measure many, many laboratory values and tests, how can you get them? So this is a limitation. But it's very important to serve as a baseline for future studies. And this is why it's critical to conduct a natural history study to see if you have a new therapy or if you're seeing something, a new complication of the disorder, how to define it. It requires a tremendous amount of teamwork between the patients, the care providers, and then the research teams. And at this point, if anyone's listening online, I would express my great appreciation for all the patient families who come to visit us. And it mainly focuses on the well state. Understanding what happens when someone is decompensated or has hyperammonemia or they're having an acute cardiac event, that's very, very important, but it's typically not part of the baseline natural history study. You would record that and you would want to get more information about it, but it's not necessarily part of what we've been doing at the NIH. So here's an example of when someone enters a natural history study. I'm showing you a picture of a heart. I'm not going to say much more. We have an expert cardiologist here to talk about echoes and EKGs. Is a picture of a patient getting an echo, and every patient would come in. It doesn't matter if they have symptoms or not. This is part of a natural history study. You would get an echo no matter what. And when we do this, sometimes we discover a patient has an issue with their heart or their kidney or whatever we're imaging. But it's part of a protocol, so everyone gets it. So when patients come to visit us, we collect about 500 parameters per visit over 2 to 5 day stay. And you can see here what happens, how it's broken down. And this data is aggregated. We have our own databases that has this medical information in it. Then we actually mine those databases. And I'm going to give you an example, by the way, Dr. Shlachkov prepared this slide, of what you can do with such information. Oh, actually, also to say this. This just is a picture of the patients that are enrolled in our study. We're quite fortunate and again, gracious that all the families have participated. I can't really read the pie chart from the back. I'm looking at a screen that's very small, but you can see how many patients we have, and it's about equal 50-50 PCC A and B. And we split it out by how many have transplants and how many don't. And there's lots more information we have about the molecular genetics and other information that I won't go through. Okay, so that's just a bit about natural history. That's part of research. And so from that, you can collect really important information that can help identify new complications, perhaps in the patients, and give information about monitoring. And I'm going to go over an example of that right now. One of the things that can happen to patients, and it's understudied, is chronic renal disease, CKD, chronic kidney disease. It's part of MMA. It's very, very common. So part of the reason I keep talking about MMA, but methylmalonic acidemia is a very close condition to propionic acidemia, and we study both. The question is are kidney problems common in propionic acidemia? If so, who's at risk for them, and what do we do to screen for them? And then the treatment is something separate. So what I'm showing you here is just measurements we've made of a biomarker of the kidney. Many, many patients. Each one is different. But when you put them together, all of a sudden you see a pattern. That kidney function can decline with age in PA patients. And it can go down very low to even to the level where some people need a kidney transplant. This was not known until we did our study. Why is it important? Well, here's some reasons why. You can put them on the screen. It has implications for giving medicine. It has implications for new therapeutics. It has implications for monitoring. It has implications for study design. And then, of course, treatment. The last thing, sorry, it's too small. I don't know what the last thing. Yeah, medication doses. And this, if anyone wants to really read more about this, we wrote an entire paper on this. It's published. Anybody can get it if you have internet. I know some people don't have internet access, but if you just use that internet link, or we can print it out and send it to your clinic, you can read about this. But in this paper, we talk about how we study this and we make recommendations for monitoring. So for people that come to the NIH to visit us, some of the things we do is we measure kidney size in everybody, and we measure renal biomarkers, chemicals in the blood that are changing up and down with kidney function. And we have some that we measure clinically, and we have ones that we measure in the research setting. And some of the ones on the research side are better, we're finding, than the ones that you can just order as part of routine labs. Okay, next I'm going to talk about biomarkers, predictive biomarkers in PA. And this is going to be, I'm just going to warn everybody, this one's going to be like much harder scientifically. It's going to be a little harder to follow for possibly. What is a biomarker? Let's just start off with that. This is just a generic definition. Not everybody would agree with this. It's a molecule found in the blood or other body fluid that's a sign of the condition. Usually it's elevated in a condition or low. It goes with the severity of the disorder. It can be used to see how well someone responds to a treatment. And the major question some people would have is, could you use it as a clinical outcome? In other words, you give a patient gets a new medicine or a treatment and this biomarker changes, and then you can say it really worked. It's making their condition better. Unfortunately, that's just not the case. You have to prove that. It's not so easy to prove. It makes a lot of sense, but it's not easy to prove. So there's a motivation for this, a really important one, that there's no FDA-approved response biomarkers I'm aware of for organic acidemias. And with the development of, at least in my group, of gene therapeutics, this is really important. The diagnostic biomarkers, the ones we think about, those are good candidates, okay? MMA level, propionyl-carnitine level, 3-hydroxypropionate level, methyl citrate level, these are chemicals that you would measure. There's a serious problem with this, though, which is when someone gets a liver or liver kidney transplant, those levels stay very high. They stay very high. And there's a reason why from the metabolism side, related to something called obligative oxidation, which you will never get rid of this. Every cell could potentially make organic acids. And the reason we know this is absolutely the case. You want to switch that down? Okay. Okay. This is working here. Let's see. Okay. There we go. That's better. I was talking about the production, what we'll call the extra hepatorenal production of metabolites. This happens. And the reason we know this is really significant, and it caused a lot of confusion in the field, I would say originally, is because when patients, and I'm going to talk about methylmalonic acidemia here first, received liver kidney transplants, and everyone said the patient is cured. They went and then measured the MMA level in the blood. It was still 1,000-fold elevated. But the patient was doing clinically well. See? So that's the problem with some of the biomarkers. In PA, the same thing can happen with methyl citrate. A patient that has a liver or liver kidney transplant with propionic acidemia will have methylcitratemia and methylcitraturia. So if it was so effective, why did the metabolites still so high? This is a conundrum. So how do we solve this? That suggests we might need alternative biomarkers. I'm going to talk about one for the liver. There's something else which I'm going to go through, and this is a harder one, but I'm going to go through this about how we got there. And it started with MMA, again, MMA mice in a gene therapy study we did many years ago. So the way this method I'm going to talk about works is the patient or the mouse receives a non-radioactive heavy carbon trace. You either drink it, it goes into the G tube, or the mouse gets injected into their belly. And if the enzymes are working in the liver, you can see the little cartoon person over here breathing out this gas. You measure the gas and you see if heavy carbon dioxide is in the gas. That means that the enzyme that's missing from the person actually is in their liver and it's working. Okay? So the way we started this, and again, this is just to say a word, I'm showing you a picture of a little mouse over here. It's a sick little mouse, has methylmalonic acidemia. That's what the mice look like when you don't treat them, if you try to keep them alive. And I should say to you that I know people here might work with animals. We check the mice at least twice a day, sometimes 3 times a day. They get fluids, they get hot water, heating pads, special diets, amazing. We take amazing care of the mice, but if they have a really severe condition like an MMA problem, this is what happens. This is natural history. They're sick and they die. Now, when we did gene therapy on the mice, we converted the mice from those sick little mice up in you can see the scruffy little mouse, and they looked like that. You couldn't tell the difference. They looked completely normal. We said, we cured the mice. It's amazing. It worked. The problem was this. When we measured their MMA levels, they were 1,000-fold elevated. Just like patients who received a liver kidney transplant, they have very high MMA levels. How could that be? How come they're doing so well? Well, the answer is because they can oxidize propionic acid. And this is how the assay works. There's Dr. Manoli, my good colleague. So in the mice and in the patients, we give them this tracer, and then we measure in this little chamber devices that we've developed to see if they can exhale the gas, and you measure it. And you can do the same thing on a patient. And in a patient, what you do first is you measure their RQ. You measure the patient's gas release. It doesn't hurt. This is non-invasive. And then you collect their breath after they drink the heavy carbon, and you see if it works. So there's the mice. That's we got our answer. If you look in the middle, that middle line, that shows the oxidation. The bottom line shows a mouse that doesn't get the therapy. They're not oxidizing. The middle line shows the mice 1 year after gene therapy. They're releasing label. And then the top line shows the control. So you can see why those mice that I showed you before that have the high level, why they're doing well. It's because they can oxidize. What about patients? So this is what we do in patients, and this is how it works. They get this metabolic cart study where we make measurements because you need to know something called the VCO2. And then we give them this label and we see, how they breathe. It doesn't hurt, as I said. There's even a device we use where you can hook up a patient to automate this. It's a machine. It goes into their nose. You don't do anything. They just drink the label. So there's a patient, a control, that little bump if you look on your upper left, that's the gas, the kinetics. So they're releasing label quickly. Below that is a patient with methylmalonic acidemia, and you can see what's happening. They can't release the label. So we did a study, Dr. Shalachkov did, and we tried to determine if when we make these measurements, so they correlate with the patient outcomes, they correlate with clinically relevant clinical outcomes. You can see all of them listed here. The answer is yes. I'm not going to go through these graphs, but it's a really nice correlation. This is the best biomarker in our study. And this was really what is really exciting is this, and this is a harder one, but let's just go through this. If you look at the bottom lines are patients that have methylmalonic acidemia. This is the first example is MMA, okay? So you can see what's happening. The patients are not fully oxidizing. If you can look on the graph, you can see in there, it says MUT-LKT. I don't have a pointer again. But what you can see is the patients that have received the transplant are fully corrected, despite the fact they have really high metabolite levels. This test can discriminate that. And here's how it works in propionic acidemia. The same thing. The blue dots, those are the normal controls. The red, those are patients with propionic acidemia. And I should say, this test sort of reflects enzyme level activity in the patient's body, mainly in their liver. So some patients can have higher activity. That's why they look almost normal. Some of them are actually normal on this test, but they definitely have propionic acidemia. Most patients, however, are much lower. And so if you see those patients in the red at the bottom, after they get a liver transplant, you see what happens to them. That's the green. They go to abnormal or even above normal. And one of the reasons that is because some of those patients have received very large livers. So when the surgeons, I see one of my surgical colleagues over there, they tend to get the biggest liver that you can put in that fits nicely. And so those patients have more liver. That's why they're oxidizing better. Okay. So that's the message. After you do this in patients, this measurement shows the effect in the liver, despite the metabolites. So this is what you want to use to see if a liver-directed treatment works. This, not biomarkers. Per se, you need to use all of them, but this one, it helps. I'm going to skip over this just to talk about the analytic performance, how the assay works, but just to show that, again, many patients are very low, some patients are variable. I think we have in the middle slides coming up here in a minute to show that. I think I showed part of that. Anyways, let's not focus on this. Just really a lot of details about how this test performs as a test. And there's even more on this, how it correlates. So this is something else what some people would want to know. If a person has really severe changes in the gene, what happens to their oxidation? It's very low. If a person has a quote unquote milder, and by no means are we saying anybody that has Plains type or Amish variant is mild. It's not, I'm not saying that, but just enzymatically, it doesn't have the same effect. You can see what happens. We have one patient, and I'll say that this family came to see us. I think they took a very long cab ride, perhaps, to see us at the NIH. And they participated in the study. That's the little green dot. You can see there, quote unquote, 2 Amish PCCB alleles. That's what we're calling it. And then, but if a person has the same change that's seen in the Plains population, then on the other chromosome has a different change, you can see what happens. If it's a more severe change enzymatically, they have less of an ability to oxidize label. So for those who are really interested, this would be amazing if anyone wants to look these up and read them, but this is an incredibly detailed papers we wrote. And I'm going to just make a mention about something of an advanced technique in this last paper that I show up here. It says BioWolf on it. And this is a paper that Dr. Shalachkov led and developed this, where what he did was develop a machine learning algorithm that used all of our NIH data from our patients and ran on a supercomputer, then asked the computer, with all this information, what do you pick? What does the computer pick? And the computer will pick, there's the ways a computer will do things. It will do every single permutation. And the computer ended up picking propionic oxidation as the #1 predictive biomarker of severity in the condition. And then a number of other ones that were really interesting that we would never have figured out, but the computer did. And so we went back and looked at those, and they're quite accurate, actually. Really great paper if anyone wants to really get into it and read that one. Okay, finally, we're going to finish off on gene therapy. Okay, this is going to be slightly technical. So I'm going to start off by just having some real basic concepts of what is gene therapy. And this is, again, a generic definition. It's the transfer of genetic material into a patient or their cells. And there's different reasons you do this. You do it to introduce the gene. Okay, that's simple. That's gene addition therapy. You can actually do this to try to correct the gene in a patient, in their body or their cells. Or you can actually do a gene therapy to inactivate a gene. If that gene is causing a problem, you can inactivate it. So those are sort of the big baskets. Is this new? A new idea? No. It's a very old idea. It came out around the time when we were first, as a community, able to clone genes. And you can see here, it was actually done in 2 children, not with PA, in the 1970s. And I've showed you a little picture there using what these children had, something called arginase deficiency, which is a type of a urea cycle disorder. And they were given a peculiar type of a virus because someone saw something in a cell that maybe suggested it worked. And it created quite a bit of controversy and reassessment of the field that's constantly been ongoing. But just the idea of doing this is very, very, very old. There's even other old examples. So as I go through this, I'm just going to talk about vocabulary a little bit here. What is a vector? A vector is a name for a carrier of the therapeutic gene. I probably should have put a horse and buggy here for people to think it as a carrier. Okay, I have a car, but that's how it's going to get into the cell. Okay, the transgene, that's what's inside. So that's like the passenger. And then transduction is another word that means getting that cargo inside the cell. Transduction, okay, and unloading it. So as we develop gene therapeutics for any condition, this is sort of just, if you could pick any characteristics, these are some of the things you would probably like in your therapeutic. You can control the administration by injection, preferably a one-time injection. You want it to go to your desired tissues and into specific cells. It's not just enough to go, say, into the liver. The liver has a lot of different cells in it. You want it maybe to go right only into the hepatocyte. That's the main cell of the liver. You want it to be stable. You don't have to want to keep doing it over and over again. You want a stable effect, and you don't want any toxicity. This is ideal. Most things are not ideal in this world, including any type of new medicine. We and others have used something called adeno-associated virus. Okay, an adeno-associated virus is what's called a dependovirus. On its own, it can't do anything because it can't replicate. It's a dependovirus. And it's thought to be non-pathogenic. Now, when this type of a virus is reconfigured, so think of what you would, if it was a virus, if you took everything out of the virus and put in your favorite material and got rid of almost everything out of the virus, that's what you do with this type of a virus. You use it to deliver the transgene. And when that's done, it can work to express the transgene for years and years and years in animals, in dogs and monkeys. 5 years, 7 years even. It's used in humans in clinical trials. And there's even 5 FDA-approved therapies that use types of AAVs, including one called Zolgensma. Let me go back. And you can see here, there's different coatings of the virus. This is where it's going to get a little complicated. So-called serotypes. And let me try to explain serotype in a fashion that maybe people can relate to, which is this. I don't know. Does everyone here get the flu shot? Do we know? Okay. How about a common cold? People have colds, right? When a person gets exposed to one virus, they tend to get immune to it, all right? And then if a new virus comes along and you're immune to sort of something that's very close to it, you'll be immune to that virus, right? So the way that these AAVs work is they have different outer coatings on them. And that's what's the so-called serotypes. And the serotype 9 is the one we're using. So now with the experiments we've done is to make genetically engineered mice that have PCCA deficiency. We also have mice that have PCCB deficiency. Then we inject the mice with this virus and we do things like look at their survival, their phenotype, how the biomarkers changed, whether they express the transgene, where the gene went, et cetera, okay? Goal, target, as we heard from our previous cardiologist speaker, liver and heart, both. So we do what one does at first when you do this is you use different doses. And that's what I'm showing you here. This is maybe harder to see, but those little lines are the different doses of the virus. So low dose, it doesn't work. Medium dose, it works. High dose, 2 high doses seems like it works quite well. It keeps the mice alive for almost 100 days. And this is an early development. We have updated curves that are even better than this. Basically, using this data with NCATS as our sponsor, we were able to get a special designation for this. Now, when the mice get the therapy, now one of the things we do is to see if they can oxidize label. Remember that oxidation study I was telling you about? They can oxidize label just beautifully. And then with mice, you can do things like more studies. You can sacrifice them and study their tissues. And you can see here, what I'm showing you here is the pictures show that the dark red, that's the gene being expressed in the liver and the heart. And then you can actually do something else, which is you can take the hearts and the livers out and you can measure the actual enzyme. And what you're seeing here is these green bands at the bottom, that says cardiac, that's the enzyme being expressed in the hearts of the mice. And then above, that's the liver. And you can see where there's a space there where there's nothing being expressed. Those are knockout mice. They don't have any enzyme. That's why they die. So this treatment goes to the liver and the heart, and it's stable. What about PCCB? If anybody here was ready to go to Rome next week, Dr. Chandler and my group is going to present similar studies, not exactly the same as this, done with PCCB, a new gene therapy, and 5 new mouse models we've made on PCCB, showing how you can get gene therapy to work in that mouse. So how would this work for patients? This is really what matters, right? How would this work for someone that wants to get it? And this is a schematic I want to acknowledge my funding sources from the National Center for Advancing Translation Sciences, that's NCATS, to something called the Platform Vector Gene Therapy Project, that's supporting our PCCA effort, and through NCATS and the Bespoke Gene Therapy Consortium, the PCCB effort. So the way this would work is the patient would come in, that you need to know if you have A or B type. Then you would get, the person would get an IV infusion. It's a single IV. There's more to say about this, what else do you have to do? But eventually what will happen is this vector, this AAV9, will go into their body. It's going to land in the liver. It's going to express the gene in the liver. It's also going to express the gene in the heart at the same time. And that's it. One so-called one and done therapy. This is experimental. We're not there yet. It's not being offered to patients. And when it is, it's going to be a phase 1-2 study. And what that means is phase 1 is safety. Safety, safety, safety. That's all that matters in phase 1. And then the second part, phase 2, is efficacy. So this is where the idea about biomarkers and outcomes becomes very, very important. Because if something was safe, then the next thing someone would want to know about, obviously, is how safe is it, but then how efficacious is it? And so that's why you need to have this long discussion I was having with you about biomarkers. So we'll, of course, be using isotopes to measure the efficacy as one of the things, a number of other biomarkers as well. But for closing remarks, I would just, to let people know, we have developed potent AAV gene therapeutics for MMA, PA, and even for cobalamin C, this other disorder. And some are going to approach the clinic. And that, I didn't really talk much about this, but you really always, for researchers and those out that are more on the technical side, you have to really make sure these experimental reagents are tested in a really rigorous fashion in the laboratory, and they're very, very accurate models to make sure they work. And you need to benchmark them against other treatments. And this is something that sometimes not everyone wants to benchmark a treatment. In other words, compare it to something else, another type of a treatment. Some people just want to test their treatment only and let someone else test the other one. But it's important to benchmark. And of course, partnerships. So I do want to close here with to say that I've always been quite grateful to the families that helped me start on this research I've been working on. And I can also tell you a story that when I started getting interested in working on organic acidemias many years ago, I went to some very senior professors at the University of Pennsylvania, at the Children's Hospital of Philadelphia, and they told me, no, don't do that, work on urea cycle disorders. They discouraged me from working in this space. And it was the families that really helped me and convinced me to continue work in this area. And a number of groups have supported the work, including PAF and people online and some of the people in the audience. So with that, I'll pause and end here. And thank you.